Hey guys, welcome back to another lecture video for Chem 104. In this lecture video, we are going to talk about polysaccharides. And so polysaccharides are a class of carbohydrates that's composed of many monosaccharide units. And so all of these monosaccharide units are going to be covalently bonded to form, this large polymer called polysaccharide. And so the name of the bond that holds these monosaccharides together is called a glycosidic bond or glycosidic linkage. And so glycosidic bonds and glycosidic linkages was formed through a dehydration reaction. Hydrolysis, on the other hand, is the opposite of a dehydration reaction. So instead of forming those covalent bonds, hydrolysis will break those covalent bonds, will break those glycosidic linkages. And this is done through exposing a polysaccharide with an amylase enzyme. Or if this is done in lab, we can take this polysaccharide and put it in an acidic solution. And so hydrolysis of a polysaccharide will give back its constituent monomers. Now, if the polysaccharide is not fully digested, then what you get is an oligosaccharide. And so oligosaccharide is another class of sugar, of carbohydrates. That's about 3 to 10 monosaccharide units. And so oligosaccharides, they do have specific functions in the cell. Primarily, they're used for cell recognition. However, in the context of what I'm describing or how I'm using oligosaccharides, is just going to be chunks of polysaccharides that are not completely digested. However, oligosaccharides do have a strong role in cell recognition. And so overall, the structure of polysaccharides, they come in two flavors. They're either linear or highly branched. And so what we're going to do is we're going to talk about the three common polysaccharides: starch, cellulose, and glycogen. And we're going to see how their structures and composition is similar to each other and how they're different as well. So the first polysaccharide that I wanted to discuss is starch. And so starch is composed of amylose and amylopectin. And starch is commonly found in specific foods such as rice and potatoes. And that's only to name a few. There's so many examples out there. Let's just stick with a common example. And so starch is found in rice and in potatoes, and starch in itself is composed of two polysaccharides. One, amylose, and number two, amylopectin. And so it turns out 20% of starch consists of amylose, and amylose itself is composed of glucose monomers, about 250 to 4,000 glucose monomers. And so you guys can imagine that if a glucose monomer was like a mini marshmallow, and if you have a bag of mini marshmallows, and if you line them all up side by side, you get like this long chain. And that long chain is going to be amylose. And so if I can draw your attention to, the image on the top, this yellow structure, all of these, hexagons that's on this yellow chain is going to represent the glucose subunit, and that, and the bonding of all those glucose subunits is basically side to side. And overall, it's going to form a linear structure. And the only reason why you guys are seeing it as a coiled structure is to increase the compactness of amylose. However, if you guys can imagine taking the ends of this helical structure as if it was like the ends of a string, and if you guys were to, pull it in either direction, then this helical structure will become linear in shape. And so overall, amylose is linear. It's taking on a helical shape to increase its compactness. Remember this, there's about 4,000 plus glucose monomers, right? So we want to make sure that it can fit inside of a cell. And so if we kind of zoom in here, we see that the linkage of the glycosidic bond is alpha 1,4. And so if you recall, alpha means that the hydroxyl group on the anomeric carbon is pointing down, right? So the OH of the anomeric carbon is pointing down. That's what's going to give us alpha. And so the covalent bond is between the anomeric carbon, carbon number 1 of one glucose molecule, and carbon number 4 of the other glucose molecule. Now, if you guys are asking yourself, okay, well, how do I know what my anomeric carbon is? Remember that you guys can always use this oxygen and this achiral carbon with a hydroxyl group as a reference point. If you recall from the Haworth structure lecture video, this achiral carbon is covalently bonded to the last chiral carbon. So this last chiral carbon is not our anomeric carbon. And so the only other carbon that this oxygen is bonded to is this carbon right here. And this carbon is going to be the anomeric carbon. And if you guys recall, the anomeric carbon used to be the carbonyl carbon of the aldehyde or the ketone. All right, and so we can go ahead and distinguish the ends of an amylose. So we have a non-reducing end and we have a reducing end. And it's very easy to identify the non-reducing and reducing end just because we have a straight chain. The reducing end represents the piece of that amylose in which the anomeric carbon of that last glucose subunit is still available. It can undergo another dehydration reaction, thereby extending the length of this polysaccharide. And so if I kind of zoom in here on the right hand side, hopefully you guys will understand why this is known as the reducing end. And so here, if we mark this as the anomeric carbon, notice that this OH, this hydroxyl group that's pointing downwards, has the potential to create another covalent bond with another glucose molecule. So I'm just kind of roughly drawing a glucose molecule here. And so since our anomeric carbon is found on the right side, we're going to say that this is the reducing end of this amylose polysaccharide. Now, if you guys are looking on the left end, this is the non-reducing end. And so what that's implying is that the anomeric carbon is not found on the left end. And so once again, if we're using oxygen as our reference point, this is our anomeric carbon. That's carbon number 1. This is carbon number 2. This is carbon number 3. This is carbon number 4. And so carbon number 4 doesn't really have that ability to initiate the dehydration reaction, if you will. It doesn't have that aldehyde functional group or that hemiacetal functional group that is observed in the anomeric carbon. And so carbon number 4 is just a regular hydroxyl group, nothing special about it. And so since there's really nothing special about that guy, we're going to go ahead and call it a non-reducing end. In other words, this hydroxyl group cannot add on, it cannot add, or it cannot extend the chain on from the left end, if you will. The length of the chain can only be extended if you have an anomeric carbon that's free. All right, so let's go ahead and talk about amylopectin. So amylopectin makes up 80% of starch. And so this is where our complex carbohydrate comes into play. And the reason why it's a complex carb is just because we have branching of that linear chain. And so amylopectin, very similar to amylose, it's composed of glucose monomers. And so there's a similarity. So this is a similarity between amylose and amylopectin. They're both composed of numerous glucose monomers. However, the difference between amylose and amylopectin is that amylopectin is a branched polysaccharide, and the branching occurs every 25 glucose unit, much like you guys see here in this image. Now, if we kind of zoom in, so we're going to just look at this section right here. And so we're going to understand the bonding, the name of the glycosidic bond between, two monosaccharides that are in a straight chain, and we're also going to look at it when it's branched. All right, so if we focus on this point, this is our anomeric carbon. It's to the right of the oxygen. And so notice that the oxygen is pointing down, so we know that that's alpha. And so this oxygen is coming from carbon number 1, and it's going to be covalently bonded to carbon number 4 in the next carbohydrate unit. And so overall, if we have an alpha-1,4 glycosidic bond, that's representing a link between two consecutive glucose monomers. So in other words, an alpha-1,4 does not represent a branch. So this is not a branching point. Okay. And so the branching point, the name of the glycosidic bond that describes the branching point is alpha-1,6. And so once again, we know that alpha means that the hydroxyl group on the anomeric carbon is pointing down. And this bond, so this covalent bond, is going to go from the first carbon to the sixth carbon of the other glucose molecule. Okay, so a 1,6 alpha-1,6 glycosidic bond is representing the branching point between two glucose chains. And so overall, if we take a step back and if we just try to create a cartoon image of what amylopectin looks like, it looks like this. And so there's many branching points. and that's going to allow many different types of enzymes to attach onto it, and to degrade it, if you will. And so this is an example of a complex carbohydrate. All right. And so overall, that's pretty much it for starch, amylose, and amylopectin. We have amylopectin that's it's more of a branched polysaccharide, whereas amylose is a simple linear chain. All right, so let's go ahead and talk about glycogen. So glycogen is a polymer of glucose. So once again, here we have a similarity among amylose, amylopectin, and glycogen. So it looks like glycogen is composed of many glucose subunits. However, this specific polysaccharide, it's going to be stored in the liver and in the muscle of animals. And so this glycogen is going to serve as like a resource or a reserve for glucose. And so glucose is going to be this it's going to be this carbohydrate that's going to act as our source of energy. And so if we are under starvation, if you will, then glycogen is going to be broken down into glucose units that will travel through our blood to get to all the individual cells, so that it has the resource needed to produce ATP. Okay, and so glycogen is very similar to amylopectin. However, the difference is that it's more highly branched. And so we still see, the alpha-1,4 glycosidic bonds and the alpha-1,6 glycosidic bonds. However, for this alpha-1,6 glycosidic bond, which describes the branching point, it occurs every 10 to 15 glucose units. Whereas for amylopectin, it's every 25 glucose units. And so once again, we can kind of see what all of this stuff looks like through the Haworth structure. And so the alpha-1,4 is representing, the glycosidic bond or glycosidic linkage in a straight chain. However, the alpha-1,6 is representing the branching point for glycogen. And the only difference is that between glycogen and amylopectin is the frequency of the branching points. And so this image right here, I decided to add this in just to kind of give us a image or a visual of what amylose, amylopectin, and glycogen looks like. Okay, so as you can see, glycogen is much more highly branched.